Hello, everyone. This week, several important developments in life science research highlighted a major trend that is shaping the future of biology: the shift towards integrated, multidimensional measurements of disease. In other words, researchers increasingly need to understand DNA, RNA, and protein signals together, not separately, in order to unlock new discoveries and better treatments. And that trend plays directly into the opportunity for platforms like Codetta Bio. Let me highlight a few of the most interesting breakthroughs from this past week. First, researchers published new work mapping the genetic programs that determine whether cancer fighting T cells remain effective or become exhausted. T cell exhaustion is one of the biggest barriers to successful immunotherapy. In this study, scientists identified key molecular switches that determine whether these immune cells maintain long term tumor killing activity or lose their effectiveness over time. What's important here is that discoveries like this create a new demand. Researchers now need tools that can measure immune signaling, gene expression, and protein activity simultaneously to translate these findings into real therapies. That kind of multi-omic insight is exactly where integrated measurement platforms can accelerate progress. Second, researchers introduced new tools for tracking how T cells activate and adapt across different tissues during immune responses. By studying immune cells across lymph nodes, lungs, and other organs, scientists are beginning to understand how location and molecular state influence immune function. This level of biological complexity creates a practical challenge. The more we learn about immune biology, the clearer it becomes that single dimension measurements are no longer enough. To really understand immune behavior, researchers need technologies capable of capturing multiple molecular layers from the same sample: protein markers, nucleic acids, and signaling states together. Third, there has been a continued progress in liquid biopsy and blood based disease detection. New approaches are enabling researchers to detect tumor signals in blood samples by capturing extremely low levels of circulating molecular biomarkers. The promise of liquid biopsy is enormous: earlier cancer detection, better monitoring of therapy response, and less invasive diagnostics. But the reality is that the strongest diagnostic signals often come from combinations of biomarkers, not just one molecule. That means future diagnostics will increasingly rely on high sensitivity multiplex measurement platforms capable of detecting both protein and nucleic acid signals together. Finally, another major theme this week was the expansion of single cell and atlas level biology, where researchers are mapping entire cellular ecosystems across tissues and diseases. These large data sets are revealing that disease biology is rarely driven by a single marker. Instead, it emerges from interactions between many molecular pathways and cell types. So, the next challenge in life science is not just discovering new biology, it's turning those discoveries into practical measurement tools that scientists and clinicians can use every day. And that's where Codetta Bio fits into the evolving landscape. Our goal is to help bridge the gap between complex biology discovery and real world measurements. By enabling researchers to capture multiple classes of biomarkers in a single workflow, we can help accelerate everything from translational research to clinical assay development. Because the future of life science isn't just about generating more data, it's about generating the right data in the right context at the right sensitivity. And that's the opportunity ahead. Thanks for watching, and I'll see you next week.